Good afternoon, dear students. And today's experiment, I am going to explain you how to perform a dot plot exercise by using any two sequences, maybe two protein sequences or two nucleotide sequences, which means genes or mRNA sequences, right? RNA sequences, whatever the sample is. So by this way, you can easily visualize where the two sequences get aligned exactly or where the indels means insertion deletion happens, mutations are happened in that. So this experiment is for visualizing the alignment, right, between the two sequences. So the principle is about dot plot is a graphical method because we are going to take the sequence in one sequence is in x-axis and one sequence in y-axis. So in these two, we are going to put a dot if the base of the base or amino acids, right? Base or amino acids of the in the two positions are same means we can going to put a dot in that. The dot means they both are similar. Both base pairs and bases or the amino acids are similar in the sequences. That is the meaning of that. So we can compare two sequences by identifying regions of similarity. By using this graphical method, we can easily understand how the sequences get aligned or what are the regions are aligned. So it uses sliding windows. We already knew that what is meant by sliding window approach, which means in the diagonal position of the graph, right? So specified length as word size, we can say that as a window size scored based on substitution matrix, the matrix what we can use for the alignment. In the previous experiment also, I told you that for blossom 62 maybe used for protein, and you can also use pan matrices, right? A dot is placed. This is the main thing. Dot is placed at the point where the sequences matched according to the given scoring threshold within the window. So the three things you have to remember, and one is word size, that is how much, uh, which means what is the length of the the length of the sequences to be taken for the analysis, right? And the second one is what is the matrix you are going to allow to scoring substitution matrix. This means we are going to allow this mutations. What kind of mutations are allowed? How many mutations are allowed? Like that, we can differentiate and we can choose the matrices according to the our analysis, right? Then the dot is placed and that in the class theoretical classes you can find that you can put arrow mark or hyphen, uh, right, like that. If otherwise you can put hyphen for insertion deletion or arrow marks in the diagonal or in the horizontal, like that. So it is better if you practice in a small sequence, like which means 10 bases or 10 to 15 amino acids. You just taken the 15 amino acids sequences and practice the matrix by scoring them. So you can easily understand how it's work, how the diagonal line will come uh, given a similarity. Okay. Okay. Then here and the third one is about the first one is I told you word size, window size, and the third and second one is about substitution matrix what we are going to use, and the third one is about threshold scoring. Right. This is the score. If the score is if we suppose we put a score, for example, we put 25 as a threshold score means if the matching score is increased about 25 or more than 25 means it takes as a positive and give us a result by putting the dot in that. Okay, so like this it will work. This is the principle of dot plotting. And uh, so if you want to say in single lines means we are going to see the blast result here, right? In blast we can see in the descriptive manner, and here in the blast you can do more than two sequences, right? Multiple sequential analysis and two sequences, the results also more than two. But here what happens, you can take any two sequences, you can analyze and get the results where the uh, insertion, deletion, or mutations are exact matches is there in that sequences. Okay. Applications. So visualizing similarity between different species of variants of DNA, RNA, or protein sequences. So you are going to practice in DNA, RNA, or protein sequences. In this practical, I will show you about the protein sequences, but you have to perform both the protein and DNA of your gene of interest, right? So what is the application, exact application? You can find if any inverted repeats or palindromes present in the sequences. The repeats or duplications, mutations, and consort domains, right? By this way, you can put more threshold values and window size and the what is that? And both, right? So you can increase that than the default. If you increase the threshold value and the window size means you can clearly get the console regions domains. Otherwise, you can get on more noises. Noises means you can get more dots, right? By this way, we cannot analyze by using taking in account of all the dots. So better you will understand when I am showing. Okay, materials needed a good internet connection and the two sequences in FASTA format and the bioinformatics tools. In this experiment, in this for the practical purposes, I'm going to explain you by using free web tool alone, right? There are so many tools are available. Standalone softwares are available to do this dot plot experiment, especially for the genomics. So you can, after this practical session, I will show you what are all the exclusive softwares available you can download and you can use for your projects if you are doing that means. Okay, and today we are going to use in EMBOSS. If you have time, you can use yours. And this is the web tool you can use only for nucleotides, which means DNA and RNA you can put. And here EMBOSS you can put anything, which means DNA or RNA, you can do that. Okay. So what is the procedure? Procedure. So we have to get a faster format of the sequence. For example, here this is the protein sequence. I am going to take the human hemoglobin beta subunit, and another sequences is albino mice beta subunit hemoglobin subunits. Okay, because we are doing most of the experiments in albino mice, and which will be relevant to our genome, which means all the drug, all the medicines are tested in the albino mice, then it will be used for human during the clinical trials. So we can easily compare that. For example, for in your case, you can take your gene of interest, you can compare with anything, right? So you can compare with any species, any related species, or you can choose from the blast results also to different scoring or 100% aligned sequences. You can check that how it is results will be, right? Okay, that the emboss dot that matcher is the tool, and you can just paste it or upload the files FASTA format and set parameters. So here, what is the parameters I already told you? These are the word size 10. If you are giving 10 means, it will take the 10 bases or 10 amino acids as a fragment, right? Suppose if you submit a sequence which contains more than 1000 bases or 500 amino acids means, it will first it will make a fragment of the sequence. So 10 fragments, 10 the length of the sequences will be 10. So first 10 base or the first 10 amino acids are considered as a fragment 1. Then 11th to 20th amino acid considered second fragment. Like that it will be fragmented, and threshold value if the matches. Suppose we are going to give a scoring, right? Scoring means if it matches means plus 2 or plus 5, whatever. That depends upon the matrix which we are using. So what about insertion? What about deletion? What about the matters. So we have to give the score for that. If the score is more equal or more than 23 means the dot will be placed. So that is the meaning of the threshold. And the window size is 10. So here also the sliding matching line, right? Diagonal line. The window size is 10. Actually, the 10. If you want to more console result, if I going to find a domain or find a console regions means you can increase up to 100. So if you get, depending upon the sequence, you may get more noises, right? So if the length of the sequence is increasing, gradually means the noise is also increasing. So we can increase gradually this window size and the threshold uh, score, right? If this is up to, you can put up to 75 or 50, 75 like that. This will be up to 100, right? Don't forget. Then scoring matrix, and then this EDNA field is optimally given in that, and this is E blossom 62 will be used for protein. If you are using DNA, you can use whatever the um, matrices available in the tool you can use respective to DNA or the protein. Okay. So increase the values if more noises is found. I already told you. Then click the submit button in this tool itself. Download the plot JPEG or PNG plot as an image. So you can analyze that and download the what is it perform the same for. So you can perform the same for zebrafish hemoglobin versus albino gas hemoglobin because nowadays researchers are using zebrafish as a animal to perform a basic research. So that may be an alternative to the levant, maybe, right? So you can compare the zebrafish hemoglobin and albino mice. You can also take the same species for your own gene of interest. If you are taking different organism means you can go for whatever you want, right? So again, I have take zebrafish and the albino mice. See, here in the first, I have compared human versus albino mice, and the second, what happens, I have compared that zebrafish versus albino mice. Then third, I come back to the human. No, no, zebrafish versus albino. Oh my God, I made a mistake here. Sorry, this is human versus albino mice, right? And this is actually zebrafish versus human. Don't forget this thing. I'm mistaken. I typed here. So this should be a human, right? Though so human, we have uh, find the similarity between human zebrafish, human albino. After that, we are going to find a similarity between zebrafish and albino. So these are just you can get only three plots, three figures. You can easily find that. Okay, unlike the result performing both, you have to perform both the DNA and the protein sequences. Okay. So if time permits, if you have time means you can, this is extra and optional. And you can go for your tool. This is only for DNA or RNA. So upload and adjust the scores if you want. Otherwise, leave it as a default and generate the dot plot um, figure, right, in JPEG or PNG. So how do you interpret this result? Supposing that dot plots, they, if you find any continuous diagonal line, means there will be an x and y axis. You are going to put the both sequences in the both side, which means say one sequence in x axis, one sequence in y axis. So if the sequence has got 0 to whatever the sequence length, right? From the zeroth sequence, say from the first sequence to last sequence in the rectangle, if you get the continuous diagonal line means that is highly similar, right? And then the same result if you find in the higher threshold level and the windows level means you can definitely get the region of console regions. Then here disturbs in diagonal lines. So that is discontinuous of diagonal lines means there is an insertion or deletion or mutation happens. So anti-diagonal line, which means if you are seeing in the right angle, suppose if you find the left angle means what happens? That is called as anti-diagonal line. Okay, there is inverted match or reverse complement, and here parallel lines. This is most important. Parallel lines means repeats or duplications. Okay, horizontal and vertical lines means partial matches are seen, and no dots. That means, and here in this discontinuous, right? Discontinuous will happen in the diagonal lines, which means that gaps. No dots means no similarity will be seen in that. So crisscrossing pattern. Suppose if you find X-like structures in the plotting area means those are inversions, right? So these are the principal uh, mm, applications. The we can see that we can easily find that what kind of matches or deletions or mutations or inversions in the particular places of the particular place of the two sequences, right? How they are get aligned. Okay, shall we go to the practical now? What I have to do is, so I already downloaded the FASTA format of only human. So I have to download the albino mice. Okay, I will check. Plot.